Hello and welcome back to Human Biology, Biology 100. In today's lecture, we're going to talk about biological macromolecules. So these macromolecules include things like carbohydrates. Carbohydrates include simple sugars and also polysaccharides. We're also going to talk about lipids, and lipids include things such as oils, fats, and esters. And we're going to talk about proteins because these are the building blocks for lots of different tissues in the body. Finally, we're going to finish up with a discussion of nucleic acids. Now, nucleic acids are important because these are the sort of program code that we use to build new structures in our body. All right, so when we talk about biological macromolecules, I want you to concentrate on three things. First of all, which elements are involved? Now, most of these are carbon-based, right? So they're going to have carbon and hydrogen, probably some oxygen as well. You also want to look at the ratio of different elements. Some have nitrogen in there, and some have other functional groups, which we'll talk about. So using these all three together, you should be able to identify all four of the major macromolecules in the human body. Okay, so let's talk about macromolecules. Macro means big and micro means small. So these macromolecules are based on smaller molecules called monomers. Mono here means one. So monomers are subunits that serve as the building blocks for these larger molecules called polymers. So monomers link together by a process called dehydration synthesis, and we're going to demonstrate that in just a minute. And covalent bonding occurs when these bonds are formed. And you want to ask yourself, for each of these different macromolecules, are they soluble in water? Because some are and some aren't. Okay, so let's start out with a discussion of monomer and polymers. Remember, mono means one, so we have one single subunit here, and we link those monomers together via dehydration synthesis to make something called a polymer. For example, an amino acid would be a single monomer, and a protein or a polypeptide would be a polymer. Okay, so here's just sort of like idiot's guide to how these things link together via dehydration synthesis. So synthesis means to create. So synthesis means we take two molecules, put them together to synthesize or make a larger molecule. Now, in the process, dehydration occurs. What does that mean? Well, it means loss of water. So here you can see we have two different functional groups, a hydroxyl group, and then on this side we have a hydrogen, and they bond together and they form a water molecule that's pulled off in the formation of this polymer. Okay. So here's an example of dehydration synthesis. We have two simple monosaccharides, simple sugars up here, linking together. We lose a molecule of water, and now they're linked together to form a disaccharide. Don't get freaked out with being able to recognize all the different functional groups here, but what I want you to understand is when we put two molecules together via dehydration synthesis, we create one larger molecule, and we also remove a molecule of water. Okay, so the opposite of dehydration synthesis is a process called hydrolysis. The word hydrolysis has two parts. Hydro meaning water, it's going to use some water, and lysis meaning to break apart. So hydrolysis is a catabolic reaction that breaks down larger molecules into smaller molecules with the benefit of using some water. So here we can see we have a polymer that's made of three subunits. We add a water molecule, we break it down into two smaller monomers, which are smaller than the polymer was. So the big picture here is that hydrolysis requires a molecule of water in order to break down polymers into monomers, and is the opposite process of dehydration synthesis. Okay, so here's an example of hydrolysis as far as a chemical reaction. On this side we have something called maltose, and maltose is a simple sugar. We add water to it, and we break that down into two glucose molecules, which are a little bit smaller than the maltose molecule. Remember, hydrolysis requires that we add a water molecule back in order to break these two bonds apart. Okay, so now that we've talked about dehydration synthesis and hydrolysis, let's talk about our first group of molecules, which are carbohydrates. Now, carbohydrates are things like simple sugars, starches, and other types of polysaccharides. The elements here should be no surprise. They're called carbohydrates, so it has carbon and hydrogen, but it also has some oxygen there as well. Now, most carbohydrates we're going to talk about are derived from photosynthesis, right? They're derived from a plant that made, let's say, sugar, or it made starch or something like that. So most of what we're talking about is derived from photosynthesis. The monomers here for carbohydrates are called monosaccharides, meaning simple sugars. The polymers are called polysaccharides. Poly means many, saccharide means sugars. So many sugars linked together by a dehydration synthesis. And finally, most of the carbohydrates we're going to talk about are indeed water soluble. Think about adding some sugar to your tea. It dissolves very readily. Okay, so let's talk about the functions of carbohydrates first of all. So a lot of carbohydrates can be used for energy storage. For example, the starches in plants. Now think about plants that have lots of starch, such as taro or potatoes or something like that. They're storing their excess energy in the tuber or in the root as starch, which is a polysaccharide. And then there's the plants that we really like to eat, like mangoes and stuff like that, and oranges. Mangoes and oranges store this extra energy in the fruit as something called simple sugars. So glucose, fructose, sucrose, these are all sugars. And in this case, the sugars are there not to really provide energy for the plant itself, but to hopefully entice animals in to eat the fruit so they'll poop out the seeds and spread the seeds around. Other types of carbohydrates that we find in the animal kingdom include structural components. For example, what we see here in this locust, the exoskeleton is actually made of a polysaccharide called chitin, and chitin is like this insoluble polysaccharide that helps give the skeleton some rigidity and protects the insect. The second group of polysaccharides we see in the plant kingdom is, of course, something called cellulose. And cellulose is a lignified material that gives woody plants sort of the support they need to grow very, very tall. So both of these are structural polysaccharides, and neither one of them are really digestible. Okay, let's look at examples of monosaccharides. So these are two examples here of monosaccharides. We have right here fructose and glucose. So glucose there, fructose there, both monosaccharides. Do you need to be able to draw them? No, but realize that at each one of these intersections, we're going to have a carbon in here. Carbon, 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 etc. Okay, so you should be able to recognize the fact that they're kind of like 1, 2, 3, 4, 5, 6 little corners there, and they have 1, 2, 3, 4, 5, 6 sides. Okay, disaccharides, on the other hand, are two monosaccharides linked together by dehydration synthesis. So up top here, we have lactose. Where have you heard of lactose before? Well, lactose we find in dairy products, right? We find it in cheese, we find it in milk, etc. And some of us can eat lactose, but some of us as we get older can't eat lactose. The reason lactose causes a problem is that when we digest sugars, they pretty much have to be absorbed as monosaccharides. We have to be able to break that down into two monosaccharides. Well, some people no longer have the enzyme to break that down as they get older, and so as a result, this disaccharide isn't absorbed in the intestine, continues down to the colon, large intestine, where the bacteria can digest it, and they produce lots of fun stuff like hydrogen sulfide, hydrogen gas, and methane, which causes all the discomfort that's accompanying lactose intolerance. Okay, other examples of disaccharides here we have something called maltose, and finally sucrose. Sucrose is table sugar, right? It's two monosaccharides linked together, and that makes it a little bit sweeter. And then we have the polymers. These are called polysaccharides. Remember that poly here means many, so we have many sugars, each of these six carbon compounds, 1, 2, 3, 4. So this is a polysaccharide, and it's joined together via dehydration synthesis to form something called a starch. Now think about what kinds of foods have starch in them. Things like potatoes and taro. Starch does not taste very sweet, okay? It tastes starchy, but it's still got lots of energy in it, and we can break these starches down into monosaccharides by hydrolysis. Okay, our next group of macromolecules are the lipids. Lipids include things like oils, fats, waxes, phospholipids, and steroids. The elements here involved are the same ones that were involved with carbohydrates. That is carbon, hydrogen, and oxygen. But the ratios are a little bit different. We have a lot less oxygen, a lot longer chains of carbon and hydrogen, and some of these are going to have phosphorus and nitrogen, but for the most part, they are water insoluble. They don't mix with water. So you think about taking some olive oil and mixing it with a glass of water or mixing it with some vinegar in order to make salad dressing. You can shake it up and it will temporarily kind of mix together, but very quickly it will separate out because oil and water don't mix. So that's a big thing to remember about lipids is they do not mix with water. Okay, so what are the functions of lipids? Well, there's a lot of functions. One, for people like me, it's energy storage, right? As you get older, you put on more fat in your abdominal area if you're a guy, more in the buttocks and thighs if you're a girl, but this is a form of energy storage. As we consume too many calories, we convert some of those calories into adipose tissue or fat so that we can draw upon that later on. The other thing that lipids do for animals is they add protection and cushioning to body organs. For example, around the kidneys, you have this nice capsule of fat that helps to insulate the kidneys and keep them from rolling around. They also serve as structural components for membranes. As we're going to find out in a chapter or two, the cell membrane is composed of something called phospholipids, which forms a semi-permeable barrier between the inside of the cell and the outside environment. And finally, some lipids form chemical messengers called hormones. So you've heard of steroids before. There are types of hormones called steroid hormones which are based on lipid molecules. Okay, so there's 3 different types of lipids that we need to talk about here today. The first are the triglycerides. These are the neutral fats. These are the ones you love to eat in the food that you eat. And then we have something called phospholipids found in cell membranes. And finally, the steroids, which we'll talk about, form the basis of some hormones. Okay, first let's look at something called triglycerides. Think about what that word means. Tri means 3. There's going to be 3 of something, and then a glycerol molecule. So what we can see here is at the very end of the molecule, we have this glycerol, which is going top to bottom, and then we've added 1 fatty acid tail in there. In reality, this triglyceride, once it's formed, is going to have 3 of these fatty acid tails, and these are long hydrocarbon chains, which generally make this molecule insoluble in water. Okay, so triglycerides are grouped into one of 2 types. They include saturated fats and unsaturated fats. Regardless of what type they are, both types include a glycerol molecule, which we can see up top, plus 3 fatty acid tails, which you see 1, 2, 3 right there. Okay, saturated fats are called saturated because the carbons in there are literally saturated with as many hydrogens as possible. So take a look at this triglyceride. We have a glycerol molecule up here, and then we have our 3 fatty acid tails right there. If you take a look at the carbons in each of those fatty acid tails, they're bound to 4 different atoms here. This carbon's bound to this carbon, but it's also bound to this carbon, and it's also bound to 2 different hydrogens. So they have as many bonds as they can with all 4 different atoms. So that's why they're saturated. And as a result of being saturated, they tend to be solid at room temperature. And because they're solid at room temperature, they can cause a greater risk of heart disease in people that eat too much saturated fat. So examples of saturated fat in your diet are things like cheese, which everybody loves, butter, and also animal fats. Because they're solid at room temperature, again, we have an increased risk for heart disease, which we want to avoid. So we want to try to eat a minimum of saturated fats in our diet, even though they're super tasty. Unsaturated fats, on the other hand, have 1 or more double bonds between adjacent carbons, like we can see here. So this carbon is double bonded to this carbon. It's sharing 2 pairs of electrons, and as a result, it has fewer electrons to share with hydrogens. So there's fewer hydrogens here. There's no hydrogens on this side. There's 2 hydrogens on that side. So as a result, these molecules stack together differently and tend to be liquid at room temperature. So we tend to think of things like vegetable oils as being unsaturated fats. And unsaturated fats carry a lower risk of heart disease than saturated fats. So here are some examples of unsaturated fats. We've got some Wesson oil on there, canola oil, etc. So all of these tend to be better for you than saturated fats. They still have a lot of calories. They still have 9 kilocalories per gram, so we do have to watch how much we eat, but there is a lower risk of heart disease with unsaturated fats. Now, in addition to saturated and unsaturated fats, we have something that's kind of in between called partially hydrogenated oils. So you probably read on the back of a peanut butter can or something like that, something about partially hydrogenated soybean oil. And what is that? Well, initially it was an oil, it was an unsaturated fat, but then we've gone back and actually added hydrogens to that oil to make it solid at room temperature. And so these are found in things like Oreo cookies, Crisco, which is like this, I don't know, grease-like substance made out of oil, and even some types of peanut butter. Now, the problem with these partially hydrogenated oils is they are now saturated essentially, and they may even be worse for you than some of the natural saturated fats that we find in buttermilk or cheese. Okay, our next group of lipids are something called phospholipids. Now, phospholipids aren't something that we normally include in our diet. I mean, they do come in the foods that we eat, but we tend to find them mostly in cell membranes. And these differ with triglycerides in that they only have 2 fatty acid tails. And these fatty acid tails are hydrophobic. They don't like water. But it has this unique structure up top, a head that has a phosphate ion in there, and that phosphate ion makes it hydrophilic. So we have a hydrophilic head that likes water and 2 hydrophobic tails which do not like water. And the way that these phospholipids orient in the cell membrane is they stick their heads towards the outside and inside of the cell where there's plenty of water, and the tails, which don't like water, basically intermingle with each other in between the 2 sides of the membrane and try to exclude as much water as possible. We'll talk a lot about the phospholipid membrane in the chapter on the cell, but suffice it to say, it forms a semi-permeable membrane which allows some stuff into the cell but not other stuff. Okay, our next group of molecules are something called steroids. Now, steroids is a subgroup of the lipids. It's still a lipid like a fat or an oil, but the difference here is it's based on 4 interconnected carbon rings. And this is how we should identify anything that's a steroid is going to have these 4 different rings in there. Now, some of these are going to be hormones, and we'll talk about steroid hormones, but some of them are not hormones. But all of them have these 4 different interconnected carbon rings. Now, the first steroid you need to know about is something called cholesterol. Now, cholesterol is a molecule that we get in our food. We find it associated with eggs, with shrimp, and stuff like that. And you probably already know that we don't want to get too much cholesterol in our diet because it carries an increased risk for heart disease. But what you probably didn't know is we actually manufacture the majority of the cholesterol in our bodies within our liver. So even though you might be adhering to a low cholesterol diet, you might still have higher cholesterol numbers as you get older as a result of the liver overproducing cholesterol. Okay. Of course, cholesterol can be modified to make a steroid hormone. And steroid hormones are things like estrogen and testosterone, right? You've heard of steroids before, right? That's what Sylvester Stallone and Arnold Schwarzenegger maybe used at one time to bump themselves up. So some of these hormones can be androgens. If we talk about testosterone, that definitely is associated with increased muscle mass, a more skinny build. It causes us to lose fat. On the other hand, this other hormone is estradiol or estrogen. Now, this is also a steroid hormone, but it has very different effects. If you were to take this steroid hormone, it would lead to the development of breast tissue, increased fat in the buttocks, and more female secondary sex characteristics. Okay, about cholesterol. How much should we have in our diet? Well, we want to minimize the amount of cholesterol coming in because, again, your body produces a lot of its own cholesterol. These numbers have probably changed, but you can see here we probably want our cholesterol well below 170. Triglycerides are another type of fat that we find suspended in the body, and there's certain levels for that as well. And you've heard about LDL and HDL cholesterol. So there's HDL cholesterol, which is the good cholesterol. This is the cholesterol that tends to eliminate fats from the body, whereas bad cholesterol tends to accumulate in the body, forming plaques. And so we once thought that it was just the LDL number that was important, but as we know now, it's more of a ratio of HDLs to LDLs. You want your HDLs to be high and your LDLs to be low. So as you get older, cholesterol numbers can become a problem. Most of you all are probably age where cholesterol is not something you regularly think about. But eating healthy now can ensure that your cholesterol numbers maintain in the safe zone later on. So avoiding eating lots of eggs, lots of animal fats can help reduce your cholesterol, as is exercise. And eating green leafy vegetables goes a long way too. We now know that eating lots of fiber and soluble fiber can actually help wash some of the cholesterol and lipids away from your body and reduce your cholesterol somewhat. And finally, losing weight is important too. If you're very, very obese, you tend to have higher cholesterol numbers anyway. Smoking is not good. You've already heard that from the Surgeon General, but smoking also tends to increase your cholesterol levels. We do know that moderate consumption of alcohol can slightly reduce cholesterol numbers, but you're probably better off looking at one of these measures above. And finally, if none of this works, we can then look at medications such as Lipitor in order to reduce your cholesterol burden. Okay, our next group of molecules that we're going to talk about are the proteins. Now, proteins here definitely have carbon, hydrogen, oxygen, but now we have nitrogen. So nitrogen is something we didn't really have in abundance in any of the other macromolecules. So if you see nitrogen in a molecule, you want to think, is that an amino acid or is it a protein? So the monomers here are amino acids. Amino acids are based on amino groups, NH2, and the polymers are called polypeptides or proteins. And generally, if we're talking about the individual amino acids and smaller polypeptides, they are water soluble, but of course very, very, very large proteins may be water insoluble. All right, so what are the functions of proteins in the body? Well, some of them are used as enzymes. Okay, enzymes are basically things that help to speed up metabolic reactions. For example, when we talked about breaking down a disaccharide into 2 monosaccharides, oftentimes an enzyme is involved there. And we also have structural proteins, right? Proteins maintain a lot of the structure of the body tissues. We find them in our fingernails, as in keratin, and we also find collagen in our skin, and we also find collagen in, Jell-O. So if you eat Jell-O, the thing that causes it to gel is ground up collagen that's been reconstituted with water. Where did that collagen come from? Well, it came from pig ears or beef ears and also the hooves as well. So just be aware that some of these proteins in there and a dessert we might think of as being vegan are actually animal-based. Okay, functions of proteins. We already said that some proteins are enzymes. We'll talk about that. Some of them are structurally important. For example, the collagen we find in our skin. Some are chemical messengers, such as hormones, and some can be found in antibodies in our bloodstream. Okay, so the characteristic groups of proteins, we have something called a carboxylic acid group, COOH, and then more importantly, we have something called the amino group. The amine group is NH2, and this is the way to identify whether or not something's an amino acid or a protein. If we see NH2 in it in conjunction with a carboxylic acid group, COOH, we then know it's an amino acid or a protein. And finally, the other group that's in an amino acid is something called an R group. R just here means variable. It could be one of 20 different groups to make different amino acids. So here is what a stereotypical amino acid will look like. Up here we have our COOH, and that's carboxylic acid. And then we go on this side, we have our NH2 right there, and that's going to be our amino group. And then up top right there, sorry, pointing is hard, we have our R group down here, and that's going to be the rest of the molecule. So again, just look for the NH2 or the COOH, and together these indicate we're looking at an amino acid or a protein. Okay, one thing I should point out is that proteins have a three-dimensional structure, and their structure determines their function. And so this three-dimensional structure is determined by bonding of the individual amino acids as well as folding of the molecule. So starting out on this side, we can see the individual amino acid sequence here, and then we see the initial folding, the secondary, and then we see a tertiary folding here, and finally a quaternary folding, which results in the adult structure of the protein. Again, if we disrupt these chemical bonds, we're going to disrupt the structure of the protein and then disrupt its function. So think about proteins in the body like hemoglobin, which help to carry oxygen. If we subject that hemoglobin to a different environment, we could denature some of these proteins and disrupt the bonds and render that protein useless. Okay, our last group of molecules here are called nucleic acids, and nucleic acids, as the name implies, are generally found in the nucleus of the cell. The nucleus, as we're going to find out, is the control center of the cell, and it's also the site where we generate the recipes which are used to produce proteins. So the principal elements here involved in nucleic acids: carbon, hydrogen, oxygen, what we saw before, but now nitrogen and phosphorus. Remember, we did see nitrogen in amino acids, but I think this is the first time we talked about major amounts of phosphorus. Okay, the monomers here are called nucleotides, and the polymers are called nucleic acids. Generally, nucleic acids such as DNA and RNA are water-soluble. Okay, so what do we use nucleic acids for in the body? Well, first of all, they're used as a genetic instructional set, right? You probably already know that your cell nucleus contains chromosomes, and these chromosomes contain DNA molecules. The DNA molecules include all the information to replicate an entire new you. It's also a recipe system for making proteins. So DNA is used to code for RNA, and think about DNA as being the cookbook with all the recipes, whereas RNA is a single transcript or single copied down recipe which we use to make a single type of protein. And finally, we're going to talk about a nucleic acid called ATP, which is not used in genetic instructions whatsoever, but is used as an energy carrier molecule. Okay, so what are the characteristics of nucleic acids? Well, first of all, we have a 5-carbon sugar, that's called a ribose sugar. We have a phosphate group, and we have a nitrogenous base. Well, nitrogenous here just means it contains nitrogen. That's what you have to watch out for. Okay, so here is a schematic of what a nucleotide would look like. Here we have our 5-carbon sugar. Each one of these points would be a carbon, 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 and carbon. Okay, and then we have our nitrogenous base. And then finally, at this end, we have our phosphate group. Okay, now that we've gone over the schematic of an individual nucleotide, let's talk first about those nitrogenous bases. Now, the nitrogenous bases include nitrogen, right? So we have different groups of nitrogenous bases. This first group right here are called purines, and they're made up of 2 different rings. And the next group we have are called pyrimidines, and they're made up of a single ring. You do not need to be able to identify these for an exam or anything like that, but what I point out here is that we can identify them as nitrogenous bases because they include nitrogen. The next part we have of a nucleotide, of course, is the sugar, and in general we're going to have a 5-carbon sugar called a ribose sugar. We'll have something called a deoxyribose sugar, which we have over here. And so ribose sugar we find in RNA, deoxyribose sugar we find in DNA. Okay, so altogether, a nucleotide is composed of a ribose sugar or a deoxyribose sugar, a phosphate group, and then finally our nitrogenous base. And here you can see how they're all linked together. At this end here, we have our phosphate group, we have our 5-carbon sugar, and then we have our nitrogenous base. This would be a pyrimidine, this would be a purine. It's based on 2 different rings here. So this is called a polynucleotide or nucleic acid. So the most important nucleic acid in the body, you probably already know, is DNA. And DNA is a double-stranded nucleic acid, and it includes all the genetic information to rebuild an entire new you, and also to govern the protein production process in the human body. So just like any type of nucleotide, these nucleic acids also include nucleotides composed of phosphate groups, 5 carbon sugars, and a ring-shaped nitrogenous base. Remember, the pyrimidines were a single ring and the purines were a double ring. DNA contains all the information for almost all the cell activities in the body. So it's really cool. It's like this giant cookbook that we can always go to pull out recipes in order to make new proteins and even replicate ourselves when we do sexual reproduction. Finally, the last type of nucleotide we're going to talk about has nothing to do with genetic information. It has to do with carrying energy. And this nucleotide is called ATP, and it stands for adenosine triphosphate. So let's look at the parts of the molecule. We have our 5 carbon sugar here. We have our nitrogenous base. So this looks like purine to me. And then what we have here, instead of 1 phosphate group, we have 3 phosphate groups. And what you need to know is these phosphate groups are storing potential energy here. There's energy being stored which can be released later on if we break these bonds apart. So ATP is kind of like a rechargeable battery that we use in the body. Think about how you get energy into the body, right? You eat foods like carbohydrates and fats. Well, eventually we're going to convert that energy stored in those molecules into ATP. And ATP is sort of like the rechargeable debit card we can use to fuel cellular processes. So anytime we need energy, we're going to make ATP in the mitochondria and distribute that ATP throughout the cell where it can be broken down to fuel cellular processes. Okay, and here's the process in which we can break down ATP to release energy. Remember, ATP stood for adenosine triphosphate. So we have triphosphate. We break that down into a diphosphate and a free phosphate group. So we've broken off 1 phosphate, and in the process we release energy which we can use to fuel cellular processes. Now, don't let the chemical equation freak you out. What I want you to remember is that ATP is like a charged battery, and ADP, adenosine diphosphate, is like a discharged battery. But we can always add back energy to that ADP to recharge it again. Okay, you've reached the end of the lecture on biological macromolecules. If you have any questions about the lecture, please go back and review the material. I'll be posting some pictures on La Lima of sample biological macromolecules. You should be able to identify them and also tell me what their functions are in the body. For each of the molecules, you should know the monomers as well as the polymers and also what they're used for. Until next time, see you later.